All right, ladies and gentlemen, here we are kicking off Unit 2, where we are going to be discussing or looking at, zoom in too much there, where we're going to be looking at the cell, and we're going to be focusing on cellular transport and then getting into the cellular energy and those processes. So, warning, here's your warning. Much of this chapter is covered in Bio 1 and is considered a prerequisite knowledge, okay, which is one of the reasons I wanted to do this as a flip. So that way you can zoom if you are like, I know this, then you're good to go, right? So focus on what is new to you while you review what is already familiar. That is going to vary between each and every one of you. Love this quote from Emily Dickinson. Faith is a fine invention when gentlemen can see, but microscopes are prudent in an emergency. When we actually need to see what's going on. Love microscopes. So cell biology or cytology. Remember that cyto, hopefully you've done word roots. Cyto means cell, ology is study of. Okay, for cytology, we use observations from several types of microscopes. So we're going to talk about those different types of microscopes so we can gather a total picture of how that cell is put together. For example here, I love this chart which shows the various ranges for size. We start all the way at 10 meters, and you can tell that our interval here is 10 times. So we have 10 meters down to 1 meter. Between 1 meter and 0.1, we have length of some nerve and muscle cells. We can see a chicken egg and a frog egg with the unaided eye. But then we get all the way down to 100 micrometers to 10 micrometers. This is where most plant and animal cells are going to be able to be seen underneath a light microscope. This is their normal size. Okay, 10 micrometers to 100 micrometers for all cells in plants and animals. Remember, micro means 10 to the negative 6 of 1 meter is what we're looking at here. Right underneath that, between 1 micrometer and 10 micrometers, is a mitochondria, bacteria, a nucleus. And so then we get further down. A nanometer is 10 to the negative 9th, and we can see some bacteria here. Viruses are going to be much smaller than bacteria as well. And then we can go so on to ribosomes, proteins, lipids, small molecules, and atoms. Again, you can kind of get an idea of what we can see here with a light microscope and an electron microscope. So let's talk about light microscopes, or LM for short. These use visible light to illuminate the object, and they are relatively inexpensive type of microscope. And they can examine live or dead objects, which is a huge benefit. If you examine a light microscope, you're going to see an ocular lens, which is your eyepiece. You're going to see the revolving nose piece here, which carries your objective lenses. On our light microscopes that we will be using, you should have 4 objective lenses, starting as low as 4x, and then going 10x, 40x, and then 100x for your highest level of magnification at oil immersion. We place our slide on the stage, and then of course we have our light source, which is a light bulb. There are a few techniques when using a light microscope. First, the bright field is when we just shine that beautiful bright light through. The specimen is unstained. This doesn't give a lot of contrast. Not a whole lot there that we can see. So that is actually a cheek cell. And then check out the cheek cell below it that has been stained. Same thing, we're using a light microscope. But hopefully you've done this lab before where you swabbed your cheek cell and then stained it. And then we're able to view your cheek cell underneath that light microscope. Two other techniques. One is a phase contrast. Again, here what we're trying to do is create more of depth by looking at different contrasts here, and differential interference contrast, which is when we get more of a 3D kind of image. Now, hold on. I am going to escape out of this because I lost myself again. Okay, I had to pause myself for a quick moment, but I am back. So here we go. On resolution, what is resolution? Resolution is the measure of clarity. So imagine this. You're looking up into the sky, and the ability to know, is that one bright star or is that two stars really, really close together? Well, as magnification increases, the resolution decreases. So the measurement that we use for resolution is how close can 2 points be together that we can still tell that there are 2 points and not one. Okay? That is how we find resolution. Resolution is simply this. It's clarity, guys. How well can we see it? There are two things that a microscope focuses on. Magnification, which is your ocular lens times your objective lens, and resolution. How clear is the picture going to be? As we increase magnification, we know that that picture is going to be a little bit fuzzier. Light microscopes working limits are usually 100 to 1000x. Okay. Now, limitations of a light microscope. Due to the fact that we cannot get to those high magnifications, we miss many cell structures that are beyond that magnification of the light microscope. So we need other ways to make observations. So there are some variations we have with light microscopes. First, we have some that use fluorescence, which uses dyes to make parts of the cells glow. It's oh so pretty. We use phase contrast, which enhances contrast and density, so you can get kind of more of a depth perception there. And we have confocal, which uses layers and special optics to focus only on narrow slides of cells. So let's take a look at a confocal image. On a confocal image here, this one right here we see using confocal, where the green is the nerve cell, the red are the support cells that surround it, and the yellow is the overlap between. This is what an image looks like with confocal. Without it, it's just blurry as all get out. So let's take a look at electron microscopes now. Electron microscopes use beams of electrons instead of light. They were invented in 1939, but not used much until after World War II. So that's kind of just a side note for you. Again, focus on the fact that we're using electrons. There are two types. There's a TEM, otherwise known as transmission electron microscope, and an SEM, otherwise known as a scanning electron microscope. The advantages to an electron microscope is they give us much, much higher magnifications. These magnifications are 50,000 times or higher are possible. The extreme magnification here. We can get down to the atomic level in some cases, which is huge. That's just amazing to me. Like you can see on some TEMs, I have seen the synapse, which is the juncture between two nerve cells, between the axon of one cell and the dendrite of the next. You can see the synapse. That's crazy. Disadvantages of a TEM or any electron microscope. One, you need a vacuum, and I'm not talking about a Dyson, I'm talking about a vacuum in science. Specimen must stop the electrons, and it's huge cost. These things are so expensive. We don't have one of these. Okay, and specimen preparation is another disadvantage. Now, a transmission electron microscope, or TEM, sends electrons through thinly sliced and stained specimens. It gives high magnification of interior views. This is awesome for viewing cell structures because we finally get to see the insides. The limitations of a TEM. One, the specimen has to be dead, which is not so easy when we want to keep the specimen alive. So the specimen has to be dead. The specimen preparation uses extreme chemicals, so artifacts are always a concern. Now, what's an artifact? Well, if we're using these extreme chemicals, sometimes those chemicals place an image on the specimen that wasn't supposed to be there. So it's not really a part of the cell, and that little spot is due to the chemicals from the preparation. Scanning electron microscope, or SEM. These guys are cool. I love them. They're excellent views of surfaces. They provide a great topographical image of what we are trying to look at. It produces a 3D view, and we can do live specimens sometimes as well, because you're just putting a thin layer of gold over it, running some electrons over it, and we get this fantastic image that can take those things that we don't get to see with the unaided eye, like dust mites. That's the one I always picture. That's a big SEM one that you see a lot on websites, and they show you the big dust mite. Other tools for cytology. Okay, here they are. So other tools for cytology. One is cell fractionation. This is when we break the cell apart and separate out the pieces. Chromatography is when we separate mixtures based on their solubility. And electrophoresis, which separates mixtures of protein or DNA using gels and electricity. Basic definitions there, guys. Cell fractionation simply is this. We take that cell, we throw it in a blender, we homogenate it, and then we put it through a centrifuge where we can separate out the components based on their density. Very cool. Chromatography, I just threw myself off. Chromatography is a technique for separating mixtures of chemicals. It separates chemicals by size or degree of attraction to the materials in the medium. There are different forms of chromatography. Paper chromatography, which you might have done before, where you made the little strip here and you might have put like a marker point there. We do this when trying to separate out pigments on a leaf, where we can watch the pigments travel up the paper. In this picture, they're showing water as a solvent, but in like pigments on leaves, we use an alcohol-based solvent to move those pigments up. So types are paper, we see gas chromatography, liquid chromatography, and then another form, which is thin layer. Last but not least, one of my favorite things is electrophoresis. We get to separate the mixture of chemicals by their movement in an electrical field. So what does that look like? As you can see in the image below, we're going to take DNA and we can band it out using electrophoresis. We use this for proteins and DNA. We will be doing electrophoresis coming to when we get to genetics and DNA, looking at like who the culprit is. Don't worry, we're not going to be using any of your DNA. We're just pretend we're using some DNA from a kit so we can get practice with using electrophoresis and identifying individuals. So that is all for our tools in cytology that we're going to be using. Again, a lot of this should have been review. Just a keynote, get you started and get you excited again to start working with some of these tools that we will be using in lab. So looking forward to it, and catch you later. Have a good night. Bye.